All right, ladies and gentlemen, we're going to stay on track. Just a few brief introductions. My name is Donnie Lum. I'm the past president of AIPG. And before I turn it over to Mr. Austin Dill, I want to thank you all for coming tonight. This is the Darwin Lecture Series. Due to COVID, we had our online meetings beginning in 2020, 21, 22, even 23. So we're back. We're very glad to be live again. We see outstanding lecturers and academicians to deliver these talks. We put a lot of time in it. We just don't take anybody off the street. We have a really good superstar here tonight. And Austin
Street. You've changed.
Logistics, cell phones off, please. Bathrooms are down the hall. There's water bottles in the back. So the lecture is going to be broadcast online on Zoom to around the state and etc. And if you have any conversations, you might want to take them outside. So I want to introduce Mr. Austin Dilla. He's Pittsburgh, PA, Penn Stater, so we need to follow and Jenny.
Be nice.
So he's going to introduce our superstar tonight. Thank you.
Thank you, Donnie. Thank you guys for coming. It's going to be a great event. I'm happy we can do this on Zoom and in person. That was the big thing this year. I said I want to have it in person. I like talking to people, especially geology people. You guys are my favorite. Just want to talk a little about what we do here at the KY section of the AIPG. Just before that, I want to get into our speaker, Dr. Claudia Johnson. Just a little bit about her. I'm not going to take up all of it. She can tell you about all the great things that she does. She's a professor at the Earth and Atmospheric Science Department, University of Indiana. Her formal education, a BA 1981, University of Colorado, Master's 1984 at the University of Colorado, PhD 1993, University of Colorado. At dinner, I learned that she did some work at Penn State, which was great. We are. Her research focus is on biotic replacement groups in tropical ecosystems, rates, magnitudes, and timing of tropical environment processes. She's authored and co-authored over 40 peer-reviewed publications. So I'll let her kind of dive more into all that stuff and all that fun stuff. Just kind of tell more about what we are and what we do here with the AIPG. So we will explore the evolution of coral reefs, past, present, and future today. Who we are. So we are a nonprofit organization promoting professional certification, essentially all things geology in the state as well as national. We want to be a part of. We want to be your resource for future geologists, current geologists, and past. Really just taking on whatever events. And I always talk with students and everything, and up and coming professionals, and just say, use this organization as a resource and make it all you want to make it. That's been my kind of drive with this. This is your 2024 exec board. I'm your president, President-elect Ben Curins, Vice President Daniel Draper, Secretary Tim Crumby, Treasurer Bill Braff, Communications Coordinator Richard Smath, past President Donnie Lum. This is my second full year in. I was fortunate enough to kind of get in and learn under Donnie. These guys, they have the passion, they have the years, they have the expertise. It's been really great to do some really cool events, plan some really cool events, and continue to bring all of our rock knowledge to the state of Kentucky. So here's some old events that we did. We co-hosted the 2023 meeting in Covington, Kentucky. If anybody went, it was a great event. I was one of the speakers. I talked on geophysics, which if you get me talking about geophysics, good luck. I hope you like it. We also have our course in the Bahamas, May 2023, which Mr. Frank back there leads it. Is a great course. I didn't get to go, sadly, but I hear it's a great one. We also have an outreach to all of the universities within the state here, and that's something that we continue to do. Outreach is something that I find to be very important as a young professional, having those outlets of those professionals to say, this is what I did. These are the type of opportunities out here. Let's host events of like-minded people was very important to me. Involvement with professional geologists with the KY Board of Professional Registration. That's something that I am on, as well as Bill Brab, wherever he is, I saw him. And that's just another outlet of continuing on speaking out for our profession of geology, to make sure that it's a great future for not only us, but the people that come up behind us. And just geology-focused networking events, kind of like this one here today. If you guys never met somebody, go up to them and introduce yourself, chit-chat. That's what we're all here for. And lunch and learn meetings as well. That's something I do a lot with my space of work, with geophysics presentations, really telling people about it, which, surprise, not a whole lot of people know about geophysics outside of geology. But it is helpful. It's useful. All the stuff we do and all the knowledge that we love makes a really big impact, not only here in our community, but around the entire U.S. A little tidbit, 2024 national meeting will be held in Colorado. If you can go, please go. Been out to Colorado a couple of times. I love it. If I didn't love Lexington so much, I would probably live out there. That's really all I got. I'm going to put it over to Dr. Claudia Johnson, and I'll let her take the reins.
I joined a Zoom meeting for extra credit.
Well, good evening, everybody, and thank you for inviting me to give this Darwin lecture today. I'm more thrilled than I can tell you. And I would like to say thank you very much for the Kentucky section of the American Institute of Professional Geologists. And thank you for that overview of what AIPG does. And you have a warm, wonderful group here. Very, very nice to talk to many of you before this has started this evening. And thank you, Austin, for that very nice introduction. So at the outset of my talk, what I would like to do is acknowledge my co-authors and others who have helped over the decades with my research. I especially want to thank Dr. Erica Ellswick. She runs the Analytical Geochemistry Lab at IU. But importantly, because I work with specimens and because we have a repository of specimens at Indiana University in our Earth and Atmospheric Sciences department, I know how important repositories are. So I'm acknowledging these repositories because I'm using a lot of the specimens for my research that came from avocational collectors. And they have reposited the materials in these particular collections. And I would like to acknowledge our graphic artist, Ruth Drappo, and then my students who have contributed to this talk and the various funding agencies with which I have been associated. So as you all know, this is the Darwin Lecture, and February 12th, 1809 is the birth date of Charles Darwin. And each year, universities and museums have planned outreach events to tell the public about Darwin's contributions. And every year there is a theme, and the theme of 2024 is evolutionary biology. So as a geologist, I'm taking this theme and expanding it to evolutionary paleoecology. So in my talk, I'm going to first give an overview of what the science of evolutionary paleoecology is. And then in the next two parts, I'm going to be focusing on the evolution of reefs in the Caribbean basin. For this, I'm going to be looking at Middle Cretaceous and Late Cretaceous reefs that are built out of bivalves, rudous bivalves. And then we're going to go through the biotic transition because it occurred at the Cretaceous-Paleogene boundary. And after that boundary, the reef builders were not bivalves, but scleractinian corals as they are today. So in this part of the talk, I'm going to be talking about shallow water reefs as well as deep water reefs. And I'm talking about deep water reefs because there are very few people who know about modern deep water reefs. And I want to give everybody a sense of how important they are in terms of harboring lots of biodiversity. Because I'm a geologist, I'm going to present all of the geological methods of data collection and analysis that I've used for my middle and late Cretaceous reefs. In particular, stratigraphy, sedimentology, paleontology, biostratigraphy, biogeography, in order to get a sense of depositional environments and temporal correlations in shallow water seas. In this third part of the talk on the shallow water reefs, I'm going to be presenting information that I got from use of scuba and from taking water samples very close to the corals when I'm trying to address questions on biodiversity in our shallow water reefs in areas especially that have been decimated by thermal changes in the ocean. And in the last part with the deep water corals, my approach to data collection was downloading 60,000 occurrences from NOAA's database, cleaning up the taxonomy, using a subset of that data, which are the hard corals that are constructing the reefs, and then using machine learning in order to address the question, what has the deep water diversity been like in the last 60 years? So that's a big overview of what I'm going to be talking about today. So first, evolutionary paleoecology. It's reconstructing systems of the past and taking, as geologists normally do, a very long temporal and very broad spatial scale. And evolutionary paleoecology has contributed very fundamentally to understanding the evolutionary history of life on Earth, as probably everybody in this room knows. But maybe what you don't know is we're always trying to figure out what the key transitions in biotic evolution are. And today I'm going to present the reef constructors from corals to bivalves and back to corals again. That's the major theme I'm going to be talking about in biotic transitions. And these evolutionary paleoecology is known because we compile region upon region upon region's worth of data into a global database. And two examples of this are dinosaurs. So in evolutionary paleoecology, we're looking at the biological significance of the pattern that we're investigating, whether it's taxon specific or a non-taxon specific approach. So the most famous compilation in evolutionary paleoecology is that of dinosaurs. Here's the Tyrannosaurus rex over here. So people have been studying, of course, the claws of dinosaurs, the positions of claws on the forelimbs and on the hind limbs. We've been studying the gait of dinosaurs. We've been studying the size and the position and the serration and the recurved teeth of these carnivorous dinosaurs. We know a lot about the position of the jaw joint and the way that jaw joint works in Tyrannosaurus versus Allosaurus. And so we know a lot about the evolutionary history of dinosaurs, and we take for granted this kind of chart that we buy for our grandchildren, for our children all the time. But it's because we have pieced together these key biotic transitions that we can say, so what is there a size difference? Have the carnivorous dinosaurs increased through time in size? What was the time period when we had the largest herbivorous dinosaurs? What are the scutes on the back of the Stegosaurus? Or what is this big area on the back of the Ankylosaurus for? Well, we know and we can address a lot of these questions because of these fine scale, bit by bit analyses that add up to the science of evolutionary paleoecology to let us understand the history of our Earth, the evolutionary history of biotic transitions. So we're going to take the second example of evolutionary paleoecology with reef ecosystems in looking at the evolution of marine ecospace and food webs. So here we have modes of life, and then we have older time to youngest time. So basically, this is telling us that modes of life started off with very few of them, and then, of course, they increased through time. It's something we all know already. We know this because of these big charts and graphs. So here is this issue of ecospace development through time with a seafloor starting in the Neoproterozoic and ending only here in the Devonian. But what I'd like to draw your attention to is look at how organisms partition the space on the seafloor in the Neoproterozoic time. Look at the spacing, look at the attachment, so to speak, and we can see there in the Cambrian time with the evolutionary history of lots of other benthic organisms, there's more crowding. And then very quickly, we see that not only the benthic ecospace has been filled up, but organisms started moving into the open ocean to the various levels, planktonic and nectonic of all different sizes and shapes. And not only has the evolutionary ecologic space been filled up on and above the substrate into the water column, but also look at below the substrate too, where we see very little subsurface tearing. And then when we move through time, we have lots and lots of boring and burrowing organisms that have evolved, as well as the evolution of organisms, modes of life, food webs, etc., through time. In other words, lots and lots of ways to look at both the terrestrial environment and the marine environment when we look at evolutionary paleoecology. So too, when we are looking at and focusing in on reef ecosystems, we've got geologic time over here on the y-axis, and we also have the persistence of calcium carbonate deposition in the tropics, represented by the white with the limestone figures. And then over here, we have the major skeletal elements, the major reef constructors that are represented by these mounds. And we can see that there are extinction times that are represented on this chart as well. So we're very used to, in our modern society, talking about reefs as coral reefs. But as geologists, we all know that there are lots and lots of other constructors who have made reefs through time. And if we talk about just the corals, there are rugose and tabulate corals that have constructed the reefs until the Permo-Triassic boundary. And after that time, starting in about the middle Triassic to the late Triassic, we have the order Scleractinia, which has built the reefs and continues to build the reefs today. But notice that here are the corals in the Triassic, corals, but now here are the rudous bivalves in the Cretaceous at a time when atmospheric carbon dioxide levels were 2 to 4 times present day values. So the temperatures of the oceans were warmer as well. So the hypothesis is that rudous bivalves, heterotrophic filter feeders, were able to cope with those types of environments a little bit easier than the Scleractinian corals. Scleractinian corals have zooxanthellae in them, and it's very possible that they acquired that zooxanthellae early in their history. And so they didn't have the same feeding mechanism that the rudous bivalves did. So again, we bring this issue of food webs into focus when we're looking at the large-scale picture of reef constructors through time. So in pictorial form, as you know in this audience, because there are a lot of Paleozoic rocks in our region, we have lots of tabulate and Paleozoic reefs. And in our Mesozoic reefs, and now we're focusing on the Caribbean basin, we have coral algal, and then we have rudous bivalve dominant. But there's a biotic transition. Here in greater. And I'm going to focus on middle Cretaceous and late Cretaceous, the Puerto Rico, some limestones. And then later, when I move into the modern shallow water reefs, I'm going to focus on the island of Bonaire. But for now, we look at the island of Puerto Rico and the Cretaceous. And as geologists, we always like to know what the plate tectonic setting is. So here is the Cretaceous. On the left, we have late Albian, which is middle Cretaceous 100 million years ago. And on the right, we have Maastrichtian, and this is for 72 million years ago. So this is the Yucatan Peninsula. Here is the outline of northern South America. And then this is the outline of the Caribbean plate in the middle Cretaceous time, originating south in the southwestern part of the area between North and South America, moving relatively eastward into this expanding Caribbean Ocean. And you can see the distance it has moved from 100 million to 72 million years. Here is the eastern margin of the Caribbean plate over here and the western margin. So it is these islands that I'm looking at in the plate tectonic perspective for the middle and the late Cretaceous. And they sit right here and right here. And what I'm trying to do with my research is ask the question, are these just isolated islands with the carbonates and the rudous bivalves in them? Or what do they connect to? So this is where I start out, and I'll show you how it has developed. So first I want to say that there is this very large biotic transition. And so in this space, we have gone from coral algal to coral rudous bivalve reefs. So here's a normal bivalve over here, and we've got a little jumping red dot over there. So here's a normal bivalve. Think about oysters or clams. We have the commissure over here. These are the muscle scars right here, one valve and the other. These are all rudous bivalves, and the rudous bivalves have morphologic variation, which is extreme and is almost nothing like these normal bivalves that we're used to seeing today. So during the Cretaceous time, this was an unusual time in which, again, we had a lot of CO2, warmer temperature, lots of ecospace, apparently, for the morphological innovations to take place and develop these carbonate platforms. So here is a rudous bivalve, one valve and the lower valve here, and these are teeth that fit into the sockets of the lower valve. So if I were to look at the commissure here on the rudous bivalve and here on the modern, I'll show you where the commissure is here. It's on this, and it's around here and around here and here as we're looking at. But look at how strangely shaped all of these are. So lots of morphologic variation in this group. And they were adapted apparently to these substrates, these carbonate substrates. So this is a picture I took in Jamaica. It's in an area called the Shaw Castle Schoolyard. Here's a geologic camera for scale. So that bivalve got very large during the Cretaceous time. This one is about 6 inches high. This is the commissure here. So this one is represented by the artist here. And you can see that there's some epibions on these rudous here. And this down here is an interesting picture because it is a wall that a French farmer had built when he took the rudous bivalves from his field and made a wall out of them. So again, a paleogeographic reconstruction. Here's your Asia, North America, South America, and Africa still joined. Now we're in the late Jurassic, and rudous bivalves originated in the eastern part of the tepes in the late Jurassic, and they were dispersed through larval dispersal on surface currents into the Caribbean region, through the Atlantic and into this Caribbean region. So these arrows show the distribution of the rudous bivalves for particular time periods. So at the time that we are looking at, which is in the middle Cretaceous, they are already established in the Caribbean. So I'm going to look over here. Here is the Caribbean island chain. This is the island of Puerto Rico, expanded over here. Here's the central igneous province shown over here. But the focus of my work has been on these quadrangles right here. And I'm examining 3 limestone members noted here of these particular formations. So again, we're in the central igneous province, and here we have some limestones in the middle Cretaceous. And the outcrops look like this. The research question I'm asking about these seemingly isolated limestones are, were the bivalves living in pure carbonate environments similar to today's corals? That was always my assumption, because that's what's in the textbook. All reefs are in carbonates. So can bivalves provide refined ages for these limestone members? In other words, can we relate them temporally to the Texas Gulf Coast or to anywhere else? And are these isolated limestones correlative? Well, I just answered that question. Correlative to other geographic areas. And I'm going to be looking at the Texas Gulf Coast. So that's the stage. These are what the limestone outcrops look like. Resistant limestones, and then very weathered volcanoclastic, siliciclastic, silts, shales, and sandstones. And some of these volcanoclastic materials were volcanic conglomerates and sandstones. So you can see it's very well weathered, but I can also tell you that we're working in the tropics, and so the organics have etched the limestones such that you can see that these are gastropods that we could make out the names of. And then here down in the bottom right are etched rudous bivalves. Don't look like very much, but we worked on them and we were able to get a lot of their names. So here is the stratigraphic part for these 3 limestone lenses. This is, I think, 100 centimeters. I think that's 50 centimeters per scale. So I've done the detailed stratigraphy of these 3 units, and these are about 60 meters thick at the most, 60 and 40 meters for these 3 limestone units. And what we start off with is a lot of volcanoclastic and siliciclastic material that tends to be toward the base or the middle part of these 3 limestone units. And then the upper part consists of limestone that has volcanics in it, but it also has very thick and continuous lenses of gastropods and bivalves, rudous bivalves in them. So from those rocks, we've been able to cut and polish them, and we get some sections that are good enough that we know that this is the commissure here. This is the body cavity in which the rudous bivalve lived. There's another body cavity, and this is the area, the commissure, where it holds the teeth and the socket. But importantly, to biostratigraphy, we have to be able to have some kind of traits that are specific to naming species. And it's very hard to see down here. We have a cross section and a body cavity and an accessory cavity, and lining the edges are a lot of pores. And the splitting of these pores, whether they're single pores or bifurcating or trifurcating pores, gives us indications of the species name. So what I'm saying by all of that is these bivalves evolved very rapidly. They evolved a series of morphological traits or features that we can use to identify in stratigraphic position. So these are the names of the genera. These are the names of the species that we were able to get out of these limestone members. So what we did was we used our stratigraphic column, we put the names of the genera and the species here, and we plotted the temporal presence of each of these genera and species. And over here on the right, we have the level of identification per limestone units, and then down here, the relative abundance by family. And very fortunately, we have very large abundance of a family that is the best for biostratigraphic correlations between the Caribbean islands and the Texas Gulf Coast. So what we did was we took all of these with their names, and then we went to the Texas area where Bob Scott and others have worked extensively on the Texas limestones, the rudous bivalves, and all of this, if you know Texas stratigraphy, all of these Texas rock units are tied very tightly to ammonite zones, and they're tied to planktonic foran zones. So they have very, very good, very detailed biostratigraphic resolution. So we were able to take the names of our rudis in those isolated limestones in the Caribbean and tie them to these names here and these rock units and ultimately to the ages. So in Puerto Rico, for our 3 limestone units written down here, the red is the ages that were previously assigned. Well, with our little bit of help in biostratigraphy, we were able to extend the age of this particular unit from the early to the middle Albian, and for this unit, all the way to the end of the Albian. And this is the time when there were high sea levels and lots of warming in the Cretaceous seas, especially in the Caribbean area. We were also able to take these limestone numbers and use the volcanic and siliciclastic components to get total carbon, total organic carbon, total sulfur, and total organic carbon, total sulfur ratios. And with these, we were able to plot them up. And then for the 3 distinct limestones, we were able to say which components of them were fully marine, which ones were brackish, and which ones were hypersaline, which was a surprise to us because we thought they were going to be just completely marine. So we've got brackish conditions and we've got hypersaline conditions in these stratigraphic columns that we have developed. We've also looked at them in thin section. We saw pressure compaction features, some pyrite, pyrite framboid. So we did a pretty thorough analysis of them. And our goal, of course, was to understand the depositional environments. So along with the age, which we determined here was different for each of the 3 limestones, we drew this picture of what the depositional environment was at the time that these middle Cretaceous limestones were being deposited. So remember, I told you that the volcanic plastics were very coarse-grained all the way to sands, silts, and shales. So the coarse-grained volcanic plastics and the sands indicated to us that these volcanic sources were very close to the shoreline because in the marine component, we were seeing coarse-grained volcanic plastics. So we put the shoreline very close to the volcanoes. And then, of course, we had rivers taking the finer components, bringing them out into the ocean. So in terms of biostratigraphy, we knew that all of these were about coexisting. So here's 1, the 2nd, and the 3rd limestone unit. So these are the limestone units, and these are the volcanic plastics and siliciclastics that are surrounding them. But then if we take it in time too, we have the middle Albian, and we only have 2 of the limestones, which are shown here and here. The other one dropped out, it no longer exists. And then the same thing, so we go up in time, and we only have 1 of the limestones that is persisting. So we start off with these limestones that were resistant and smashed in between lots of volcanic plastics and siliciclastics, and we are able to come up with this nice reconstruction of what they are. And importantly, for temporal correlation, we're able to tie this middle Cretaceous in here in Puerto Rico up into the Texas Gulf Coast with this dot here. Now we've done that for the middle Cretaceous, and we also were doing it for the late Cretaceous. So we moved over from Texas over to Mississippi and Alabama. And in Puerto Rico, we moved to a younger set of limestones. So we tried to tie all of that together. So these are mostly the chalks that are occurring in the late Cretaceous, when the late Cretaceous seas extended up into this area of the United States. So all of this was shallow water seaway extending down into the Caribbean. So we looked at a lot of the fossils from the Texas Gulf Coast. We also looked at the fossils from the island of Puerto Rico. Again, the rudis bivalves, we cut them, we sliced them, we worked very hard to identify the features. So this would be the body cavity, a very large rudis bivalve. Some of these bivalves were this size. at that time in the late Cretaceous prior to their extinction. And we were able to look at these, identify the genera and species, and we found out that we had this genus and species right up here, and we had this genus and species down here. And these are diagnostic fossils all over the Caribbean for the late Campanian and the Maastrichtian. So now we have a tie line between Texas and the island of Puerto Rico in the late Cretaceous time. We not only found these bivalves, but we were also able to make more of a complete depositional environment because we have inoceramid bivalves and we have oysters, and we also have a deep water sponge. So we're able to make a reconstruction from shallow water to deep water for the late Cretaceous limestones. And what that gives us then in terms of biogeography is a trail from the Caribbean. Sorry, I don't know what that dot is, but it gives us a trail from the Caribbean to Mississippi and Alabama, which is the dot right there. Okay? So we're able to tie temporally from the Caribbean these isolated limestones up into the Texas Gulf Coast. So think about it, one at a time when sea levels were high, here's the Caribbean, seas extended all the way here and all the way into the western interior, and of course connecting with these seas coming from the north as well. So there are rudous bivalves found all the way here, found in Texas, found in the Gulf Coast, and they're found in various states in the western interior seaway. But always to orient us, this is the plate tectonic reconstruction, and this is really what we're doing, connecting these areas on the eastern margin of the Caribbean plate to the Texas Gulf Coast. So the summary of the first part of this is we know that rudous bivalves originated in the Tepies and they expanded into the Caribbean on surface currents because they're bivalves and they get dispersed by larvae. And no, they weren't in plain carbonates as we're taught in the textbooks that all reefs are in. They were in mixed carbonate and volcanoplastic and siliciclastic depositional environments, because there was not one carbonate that I ever saw that was pure carbonate. It always had siliciclastic or volcanoclastic material in it. And these limestones extended through normal marine and the extremes. And importantly, rudous are biostratigraphic markers, and they're mixed in and correlated biostratigraphically to other macrofossils and other microfossils, thus providing a timeline for us to understand the Cretaceous ocean climate system a little bit better. So in this kind of reconstruction, here is the Cretaceous, and here are the rudous that we have been talking about. But if we move this way, we move across the Cretaceous Paleogene extinction boundary to the point where we only have coral and coral algal reefs. In other words, we've dropped out all of these bivalve reef constructors and all of the ammonites that were visiting the reefs. So our evidence of the Cretaceous Paleogene extinction is here, and we've come across here with the rudous bivalves. Now we're going to cross the boundary into the corals. And corals feed completely differently than the bivalves do, as I mentioned earlier in the talk. So corals are an animal. They're tissue grade of organization. So a lower grade of organization is sponge, and sponges are only cells together. Corals are the next level, cells organized into tissues. And then the next level beyond that is what the bivalves are. And they are organs and organ systems. So heart, liver, kidneys, lungs, etc. So the bivalves are a fundamentally different organizational level of evolutionary development than are the corals. The corals are the things that seem to be able to persist everywhere all the time. So they have a mouth, They have digestive filaments lining their stomach. They are on a calcium carbonate base. They are surrounded by a ring of tentacles. The ring of tentacles is shown here and is shown here, the mouth in the center. They have zooxanthellae. Zooxanthellae is an algal. It's a dinoflagellate. It's a protist. It's a golden brown algae. So plant, think of autotroph, think of photosynthesizers. They live in the tissues of the coral. They produce the food that the coral takes in. They produce over 95% of the food, the sugars, the photosynthate product from the algae, the zooxanthellae. There are about 8 clades of zooxanthellae. One common clade that the corals have is called clade C, but corals today also have zooxanthellae that are from 6 of the total 8 clades. So what corals can do today, the temperature gets hot or the UV radiation is too intense, the coral expels its clade of zooxanthellae and we get bleaching. When the temperature warms up, the zooxanthellae come back in, same clade or a different clade, maybe a more heat tolerant clade, and then the coral retains or has its color back again. So this is the color that I'm showing you right here in the tentacles. Okay. So that's how corals, one more thing about how corals operate. They reproduce sexually and they reproduce asexually. Sexually, they have the eggs and sperm that come out of this mouth area, and the eggs and sperm unite in the aqueous environment, the marine environment, and they then get carried by surface currents, and then they land on a particular place because of an attraction. Animals, they have hormones, they have receptors. They land in a particular place because of all of that chemistry. But the location that they land is usually a modified substrate. It's not pure rock, it's modified by microorganisms. And in the tests that people have done with seeds and discs, they found that the corals prefer to settle on substrates that are red or pink. Lots of encrusting organisms, foraminifera, sponges, others have the red and pink hues. So it's very, very, very interesting type ecosystem in which corals live. Corals also reproduce asexually. And so over here, you can see that these are the coral polyps. In asexually reproducing corals, we can have a bud here, and a bud here. They just bud, so they have all of the same genetics. So they have the same genetics, and then they have different genes because of the sexual reproduction. So here's the third part of the talk, and what I want to give is my perspective and the scientific community's perspective on the future health of the Caribbean reefs. So this is a picture that was taken by a colleague of mine in the Indo-Pacific in the 1970s. And what you can see here is that there are a lot of shapes, and shape is related to taxonomy. So that means there are a lot of different species of corals. And you can see here there are a lot of colors. And so those colors indicate the association of corals and zooxanthellae, which tells me it's a healthy reef. So this is what is considered a beautiful, beautiful reef at that time. Now what I'm going to do is contrast that to what we have now and why. We've been knowing for a long time that reefs are in trouble because of bleaching, because of lots of diseases. Diseases are attributed to thermal alterations in the ocean water, to ultraviolet radiation that is too intense. And the issue really is that in the marine environment, there are microorganisms everywhere. Plankton, zooplankton, phytoplankton, lots and lots of other microbes everywhere. They're absolutely everywhere in the billions. And that's no exaggeration, absolutely in the billions. But when there are nitrates or nitrites or phosphates that are put into the marine ecosystem, that is food for a subset of those microbes. And those microbes then have a population bloom. And if those population blooms are too close to the corals, they end up giving disease to the corals. Because corals live in a nutrient poor environment because they get all of their food from the zooxanthellae. They don't take the food from the outside. But when there are nitrates or nitrites, etc., pollutants, toxins coming into the system, then they develop the diseases. So we don't know much about these diseases, quite honestly. And as I tell my students, the reason we don't know much about the diseases is because of all of the more than 8 billion people in the world, how many of them are scientists? Of that number, how many people study chemistry, physics, biology, geology? Of that number, how many people are working in the terrestrial versus the marine versus the freshwater, working on plants, working on animals? Of that number, how many people died? How many people work on corals? Very few, quite honestly. So reefs are in trouble. Why are they in trouble further? Well, here's a graph that was developed by Hugh Goldberg, who has made some very significant fundamental contributions to our knowledge of coral reef science. And what we have on the y-axis is the deviation from today's temperature. Today's temperature is at the 0 line. What we have up here is atmospheric carbon dioxide increasing to the right, and down on the x-axis on the bottom, we have the carbonate ion concentration, which is doing exactly the opposite of the atmospheric CO2. So the prediction is that if we cross this threshold of 480 parts per million atmospheric carbon dioxide, we are not going to have reefs as we have today. We're going to have reefs not dominated by corals. Maybe we're going to go back to bivalve reefs again. Maybe we'll go back to bryozoa or brachiopod reefs again. As geologists, we all know about the history of life. It continues, it finds a way. So the reason we're going to be in trouble with this chemistry is because of pH. So atmospheric CO2 is taken up by the oceans, and whenever the hydrogen ion is given off, then we have a decrease in pH. And so we have a series of hydrogen ion in the hydrogen ion, I can't figure out how to say it, but we have pH dropping every time the hydrogen ion is given off. So that means that the calcium, Ca2+, cannot, quote, find the carbonate ion fast enough for the coral to precipitate CaCO3 at its site of precipitation on the coral animal. However, there are 2 corals that can change the pH of the waters at their site of precipitation. So when I teach my natural history of coral class, I always say, biology always finds a way. So here we have the terrible prediction of the slippery slope of extinction. So here we have healthy reefs and we have overfishing of herbivores. The reason that's important is because herbivorous fish eat the algae that grows in the shallow water reef ecosystem. It clears the space for those coral larvae to attach to the calcium carbonate substrate. So if we take away all of the herbivores, we get microturf, then we get fleshy macroalgae, and then we have a whole series of very negative consequences to the coral larvae finding a place to land. And of course, this fleshy macroalgae gives off a lot of ammonia, and it changes the chemistry of the waters as well. favoring other microbes that cause other diseases on the coral tissues. So here's what a healthy reef looks like. Those of you who have been diving, like Dave and Connie, you know that this is beautiful, beautiful Acropora palmata, and everywhere in the Caribbean were these beautiful stands of Acropora. You could not put your toe in the water without seeing these right offshore. Okay, so this is what a degraded reef looks like. This is a bleached reef, partly dead. This is the Caribbean now. See these big carbonate rocks? These are what this used to be. All of these sticks, these were not Acropora palmata, which is what this is. These were Acropora cervicornis stands, as big as this room and bigger. They were everywhere. They started dying off in the Caribbean in the 1970s. And then the diseases for the Acroporids hit the Indo-Pacific and did a lot of damage there. And then the diseases came back to the Caribbean in the 1990s and really did away with the Acroporids. However, look at this. That's a coral too. And that thing was probably growing in the shade of all of this. So I studied coral reefs. I studied them in the Caribbean. This is the distribution of the corals. Reefs are really big. They have a really big economic impact because of tourism, fisheries, because they are a nursery for many, many other organisms, and they have a great biodiversity. So we do want to preserve them because lots and lots of people live in the tropics, and lots of people depend on these shallow water reefs for their fishes, for their protein. So we go to the island of Bonaire, just off of Venezuela, and these are modern reefs. We go to Bonaire National Park, which is the first marine park in the Caribbean, started in about the 80s or the 1990s. Coral growth has been monitored there for over 40 years. There are no major rivers coming into the ocean. This is a dry island. The winds are coming from the northeast. This is the windy side of the island. This is the leeward side of the island. And these are all the dive sites that have lots and lots of corals on them. So no major rivers on this dry island. And it's also south of the main hurricane belt in the Caribbean. And the main hurricane belt goes along here, as we know and see on the news every year. So there's not much physical destruction by hurricane force wind in these reefs that I'm studying here. So we've worked with the Bonaire National Marine Park manager, who asked us to take a look at blocks and barrels which were serving as mooring anchors, because they wanted to switch from mooring anchors to auger pins. The reason is because they established these in the 1960s and the 1970s. They had old petroleum barrels that they filled with cement. These petroleum barrels came from Venezuela. They put them down in the 60s and 70s, and I'm stressing that because that indicates that there's a timeline for coral larvae growth. So all the corals that landed on here had to be after 1960s and 1970s. So here's the mooring buoy, here's the tagline. The boats were small in the 70s, 60s, and 70s. They grabbed the tagline instead of dropping the anchor. The reason they're switching to these auger pins and taking these away is because they started tying 1, 2, 3, 4 together, but the boats got bigger, so these blocks were then destroying the reefs because they were dragging across the bottom. So he said, so should I dump these into the big blue ocean, or should we keep them? And we said, let's keep them. Let's document the coral biodiversity in this former zone of the Acropora, where everything is dead around here. So we measured the blocks in the barrels, got all the metrics of them, We wanted to document the health and assess the health of the corals. So we collected water samples 10 centimeters away from the coral. And our goal was to evaluate the bioenvironmental conditions required to maintain the coral growth in the zone of decimated acrocoral palmata. So with scuba, we went down and we took pictures, we identified all of the corals, gave them names, we analyzed the species, we had all of the bioenvironmental parameters there, we analyzed cations and anions, and we really got a good picture of the bioenvironmental conditions. So our results are first that the corals grow on the edges and the sides rather than on the top of the barrels or blocks because of the current coming and whisking away those small larvae. Apparently they have a better chance of grabbing on and living here. But the water is clean, zero turbidity. Temperature is very normal for marine. This is about 27 to 30 Celsius. Fine for the tropics, normal salinity, normal everything. Okay, so the problem was we have the dead zones here, but we have lots of corals living here. So what's the issue with these corals? Well, we decided to do a deeper analysis on 10 sites that we looked at 5 different times over a period of 12 or 13 years. And we looked at the environmental conditions. So we have normal pH, normal temperature, salinity, normal sodium and chlorine. We have shallow water, and we have very good carbonate precipitation. So everything looked good in the water, except we saw that we had some phosphates, and we have nitrites, and we have nitrates above background in only 2 particular locations. So we started looking around saying, well, this is a dry island, what's going on? And then we found out that these have been the garbage dumps for the island of Bonaire for decades and decades and decades. And we can still see the traces of it in the water. So in terms of the species richness for the curated sites, starting in 2009 and then in 2022, you can see that species richness doesn't change very much. But what does change is the corals. So we've got the names of the corals. 2009 circle sizes indicate amounts or numbers of occurrences. So one important thing is we have Agaricea. Agaricea are plate corals. They tend to grow in deeper waters, and they grow outward to get the sunlight for their zooxanthellae. So one grows outward and outward to get that sunshine. So those are normally deeper waters. So we see them now colonizing shallower water, or what we're probably seeing is a little bit of relative sea level rise, and or we're seeing, because we have in a lot of places in the world ocean acidification, they call it reef flattening. So the carbonate could extend from here to here, but acidic waters coming in flatten the reef. They're calling that reef flattening. So we're getting more of a relative sea level rise than we expected from the modeling of 10 years ago. But important to our study, what are their survivors? So we look at the names of those survivors, and those names are here. They belong to this family. And importantly, they had this genetic rootstock from the Middle Jurassic to the recent. And these are the ones that are surviving here very nicely. They went through that Cretaceous Paleogene extinction, all the thermal changes that were subsequent to the KT and to now, and so they're surviving. So interesting. Now we go to the deep sea corals at the very last part of this talk. And the reason we're doing the deep sea corals is, I think I mentioned that we didn't know anything about deep sea corals, i.e. us at IU a while ago. So what we decided to do was take advantage of a front piece in Science in which 2 people wrote and said, we don't know very much about the deep sea around the Gulf of Florida, the Gulf of Mexico, i.e. in U.S.'s territorial waters in these particular ecosystems. So should we go out there and do bottom trawling for fish? Should we go out there and put more oil derricks when we don't know what's out there? Or should we document what's out there and then make a decision? And I read that in Science and I thought, wow, that's pretty interesting. Let me give it to my class. So I was teaching natural history of coral reefs. I copied that page. I put it on your exam. I said, read this from what you know about corals and everything about their ecosystem. What would you say? Would you say to preserve this after, to document it and then preserve it? Or go ahead and do the drilling, get the resources of proteins and the oil for the people? And so they did that. And they came back with very well-documented answers based on their opinions. And so I took it to my graduate students and I said, hey, what do you guys think? And they said, well, let's do a study. So we went to NOAA. We looked at, as I said at the beginning, 60,000 occurrences of deep water corals in the Caribbean region. So what I show here is a deep water coral reef, and these are the two main skeletal constructors of the deep water corals. So shallow water corals exist in 22 to 27 degrees Celsius. Deep water corals are much colder. They have a slower growth rate relative to the 155 millimeter per year growth rate of the shallow water corals with the zooxanthellae, because these deep water corals do not have zooxanthellae. They have a slower growth rate. They're very important. They're nurseries, as you can see here, but they are threatened by acidification, pollution, and bottom trawling. So our question is, how has deep sea coral biodiversity changed for the past 60 years? So we downloaded that database, we cleaned it taxonomically, we focused on the skeletal constructors, we had all the other environmental parameters, we used machine learning, random forest model, 86% accuracy in our training of the computer, and then we looked at the research results for these ecoregions. So we divided, this is a map of the United States, Gulf of Mexico here, into shallow and deep. So shallow is 0 to 200 meters, deep is 201 to 2,000. So we developed the coral occurrences in these ecoregions, but there are deep water corals all through the Caribbean, but we only use these in US waters. And our research results are the following. So for example, the northern Gulf of Mexico slope and shelf, and here's the Carolina Atlantic slope and shelf, that's how we divided things. So the colored dots are the observed observed data, and then the gray ones are our simulated data. So what we did was we looked at the simulated data and we looked at coral diversity through time. This is 1960 to 2020. So 1960 all the way until 2007 when they took a decrease in diversity, and then here they recovered. So that's 2007 when the big diversity drop occurred there. We can take another one. Here is the simulated data, and the diversity drop occurred in 2011. We take another one. Here's the simulated diversity. The diversity is maintaining, it's very high, and then there's a drop in 2010. So for all of these regions, these ecoregions around the Carolina and Gulf Coast, we see that there's a biodiversity decrease in just under 4 years in this whole region. Importantly, once there is a biodiversity decrease, you see biodiversity is high, here's the decrease, here's the establishment of the new level of biodiversity. Much, much lower than was up here. Here's the high biodiversity, and here's the reestablishment after the big diversity decrease. So these deep water reefs, even though we're not doing so much fishing and trawling, they are in trouble. And Oceans around the world in general are in trouble. Here's a map developed in 2008 where the researchers looked at 20 lines of evidence that would impact the health of our oceans. So, for example, think about deep water, transatlantic, transpacific cables. Think about shipping lines going all over the world. Think about major rivers putting pollutants in. Think about major areas of coastal habitation. They considered all of those. They made a map of the oceans. Blue is very low impact from humans, and then all the way to the red is very, very high impact. Our Caribbean area is one of the most highest impact areas here. This is England, Scotland, Wales. Over here, lots of oil production from the North Sea, and then this is Southeast Asia. Many, many people. So corals have a hard time, or all organisms in the oceans have a hard time. Not only because of all of that, because of ocean acidification. And we have a global observing network of ocean acidification monitors. Here's North America and South America and Africa. And you can see that we have global monitoring from mooring buoys, from satellites. We have them near shore, we have them deep ocean. We have ocean acidification alerts that are noted on the NOAA website all the time, and you can download that. So why do we care about ocean acidification? I care about it because of that calcium and carbonate ion and the need for the coral to use those to precipitate their shells. But society cares about it because when anything with a shell, bivalve, brachiopod, anything with a shell, anything made out of calcium carbonate, and most of the organisms that are benthic in the marine ocean are made out of calcium carbonate, the entire food web may be at risk. And that's why I started off with the food web at the beginning and the increase in ecospace utilization by benthic organisms. More than 1 billion people out of our 8 billion rely on food from the ocean as a source of protein, and many jobs and economies around the world are dependent on the oceans. But I always like to leave on a positive note. And in 2004, there was a huge tsunami that hit Indonesia. This was the year when the world learned the word tsunami. I think very few people knew about the word tsunami before that. But it was a devastating hurricane. The physical force of the wind destroyed the reefs. It destroyed everything on land. Lots of people died. It was terrible. Some marine scientists took these calcium carbonate blocks and they glued corals on here, which have a linear extension, 12 to 20 centimeters a year, so they grow pretty fast. And within 2 years, these blocks look like this. So the resilience and the recovery of corals can be quite strong. And so there are NGOs all around the world doing this with the corals, gluing them underwater. And if you ever thought about gluing underwater, it's quite a feat. There is a powder in your little bag here, and there's water all around, and you have to take that powder and mix it with a little bit of water and then glue something on really fast. It's quite something. So I continue to look at corals from many, many perspectives. So in the concluding remarks, we've explored the Caribbean reefs. Their future is uncertain, but my research questions are which species will persist and how can artificial intelligence, not machine learning, but even a more advanced form, help us in identifying the phylogenies, so that deep history of corals, that deep genetic history, and which ones are going to be able to persist in these modern climate fluctuations. So that is the end, and thank you. I'm very polite because my students are always pointing at the clock. So I'd be very happy to answer questions.
On one of your later charts, you had a chart of biodiversity of deep water corals. and it was a chart where it went from 1960 to 2020.
Yeah.
And they kept going, kept going,
Yeah,
kept going.
yeah.
And then it seemed like there was a step decline. And all of them were clustered around the same period of time, 2006, 2008, 2009. What happened right in that time that didn't happen over the past 40 years?
I know. See, our goal was to start with that. What happened once we saw those results? What happened? But we could not find any information. We couldn't find it. Was it trawling? Don't know. Because private companies do the trawling.
You know,
Because
all
you're
of
really
those
not
things presumably went on in the previous decades too, though. So what happened?
Water chemistry, we don't know. We just don't know. Yeah, I know. That sharp decline.
And there was geographic separated by continent.
Yes,
But yet it
yes.
was all clustered right
Yeah.
in that same time.
Yeah, and if you think of ocean currents too, so the currents are coming from south to north into the Gulf of Mexico,
Mhm,
and they're also coming from the main part of the Caribbean on the east coast of Florida.
mhm.
And so, but all of those areas were affected. I know. I don't know. Just don't know. Yes.
What was the source of it?
It was NOAA. So NOAA has been collecting that data for quite a long time, and they send down deep sea rovers, as you saw in that one picture. And they just found off of the coast of Carolina, what they're saying is the most diverse deep water reef that we have known. So they haven't documented that fully yet. They're in the process of documenting, but it's all on NOAA's database. But the thing about NOAA's database is it's hard as heck to get the data out of it. It's really a challenge. So what we have to do is we work with the computing scientists, professors, and they put a student on it for a whole semester to try and figure out how to download their platform. It's really hard. And so we've talked to the NOAA people about that, and that's where we're at. We're talking to them about it. Yes.
Considering coral's proximity to the coast and its biodiversity, what role would it have played in life moving onto land?
That's a very interesting question. So originally, you mean like in the early Paleozoic? So we've got the vertebrates. Well, we've got plants that moved onto land first. So here's what I can say about it is that coral reefs today have lots and lots of plants around them. They have lots and lots of grasses, and there are papers that are published on when those grasses germinate, how their seeds are dispersed, etc. So what I can say is probably in this ancient time, probably Silurian time, when the plants were moving onto land, they were from the ocean. We know that because of the chlorophyll match. So they're originally from the ocean, moved onto land through the freshwater source. So the best I can say to your answer of what role did coral reefs play in that transition to land is the plants were probably in the back reef shallow areas, and the corals were probably just a little bit deeper. So by proximity, if you want to answer it. Yes.
You showed a slide with the risk for different areas and pointed out the Caribbean and Great Britain and East Asia, which makes sense. But then your fourth thumbnail was an area of very low risk between what it looked like Australia and New Zealand, perhaps.
Yeah, that's exactly.
And that's a famous reef system, but yet it's not at the same risk. Would you hazard a guess as to why that area is doing better?
So that area is actually on the northern part of Australia, and the Great Barrier Reef is on the eastern part.
I know nothing about
Yeah.
Australian geography.
Yeah. So on the north, there's a city called Darwin. Darwin Day. Good question. And very few people live there. Very few people go and visit there. But the Great Barrier Reef is on the right-hand side.
And
On the right-hand side, on the eastern side. Sorry.
Yeah. So it's really just the amount of
It's a number of people.
population, God knows what else.
Yeah.
It was just interesting that that one area is so much.
Yeah. In the high Southern Ocean and the high Northern Ocean.
And then this isn't related, it's just a question. I read an article years ago, and I think Science News, it was a digest, not the full paper, about them using crushed pieces of coral to restart coral reefs. Is that your last slide about after the Southeastern or South Asian tsunami? It's the same principle, right? That it has to be on a carbonate to start. You can't just dump it on the ocean floor. Is that?
Yeah, that's a really good question. So we have these metal petroleum barrels, and really the coral larvae is not landing on the metal. It's landing on a seasoned substrate. So it's landing on a modified substrate. Yeah.
The substrate makes just as much difference
Yes.
as the pieces of what's landing on.
Yeah, and it's got to be non-mobile substrate because they're tissue, they're very small, they're just reasonably fertilized, and they can be torn apart pretty easily.
So if we found a good substrate this is a follow-up, to dump underneath them, do you think that is a good way to restore a reef system?
There's a lot of pros and cons to that. So there are artists who have taken on this issue of reef restoration, and they've built big clay, human-sized, 10-foot size statues, and they made beautiful art exhibits underwater. And the corals do attach there, and they're lovely, and they grow, and they're very pretty. We work on, my colleague at Indiana University, his name is Charles Beaker, and he's a shipwreck expert. And so we dive on these ships that are sunken around the island of the Dominican Republic. And we document all the corals that are growing there. So that's one other place. And the U.S. Navy has sunk ships in the Florida area. And those are one of the Florida study sites that IU has. Now, the cons to that are that we're putting all this beautiful stuff in the ocean, and the storms still come. And they're going to take all of that pretty stuff and knock it all over, and maybe knock more reefs over as well. So,
Thank you.
Yeah.
I had a question about the Asian reefs that you put in the British reefs.
Yes.
And I don't know anything about reefs other than the fact that I understand that they're filter feeders. And so it seemed unusual to me that you find a bank of filter feeding organisms adjacent in close proximity to volcanic highlands in the tropics that are probably generating considerable amounts of mud that are adding considerable turbidity to the water. What's the relationship there? Why would you find that sort of a large filter feeding reef system adjacent to an area that was likely very turbid?
You're asking really good questions, and I think the key to that is there's a lot of volcanic and silicic at the base, and there are carbonates, and then there's carbonates, silicic and volcanic plastics, and toward the top of those units, when we have the thick accumulations of bivalves, there are carbonates, and yes, there are volcanic plastics, but there are many, many fewer volcanic plastic blasts in the carbonates than lower down. And I think that's the answer. Yeah, that's a good question, though, because when I go to those outcrops and look at it, I just wonder how in the world. Yeah, exactly. Yeah.
Would there be modern analogs for any of those, like in Indonesia or places, the Tonga Trench area, some of the islands around there, that would have a lot of volcanism?
Oh, yeah. I know there was a guy who studied that, and what they did was they looked at those undersea volcanoes and the nutrients that would be coming out and the current dispersal near the reefs. And that's as far as I can tell you. I never really believed much of it. But I don't know. Yes, Frank.
I'm wondering about what's the role of other organisms? There are fish that eat corals, there are sea urchins that eat algae that take the place of corals, there are starfish that eat corals. What's the role of other organisms?
In today's reefs?
Yeah.
So in terms of the fish, there are the herbivorous fish, which eat off the algae, and then there are carnivorous fish, which come and eat the herbivorous fish. And then there are the other visitors to the reefs, like the starfish and the ophiuroids that you were naming. And then there are the dwellers on top of the corals. There are the borers, so the echinoids will sit on top and the teeth will bore into the corals and they're scurrying around at night. So let's see. In terms of water currents, there are bottom currents which are bringing the sediment, and they're scouring the bases of a lot of the corals. But the head corals have an ability to secrete mucus, and it entrains those sediment grains and then takes the sand grains off of the coral heads. Lots of microbes, and so many different kinds of microbes.
Don't red algae.
Dave, do you want to say something?
Well, I was just thinking about, you're talking about the coral triangle, the barrier reef, and there. There was a study done a number of years ago, back in way back 70s, 80s, where they sampled across the whole barrier reef from the continent out to the drop of the coral sea. And while I was over there, I had the opportunity to work on virtually the whole transect across and looking for findings. And, they revealed that they targeted certain organisms. And a lot of that stuff has finally been processed and published. But, one thing we noticed was that we went to the very outer edge, the beautiful clear water of the coral sea. And, there, the number of crinoid species and the abundance seemed to be low. When you went shoreward, what they call mid-shelf reefs, a lot of those are around high islands. There are some, but there the diversity goes up with crinoids and other things too. And then when you go all the way into the coastline, it goes down because that's where your runoff really puts. Of course, now they find out agricultural runoff doesn't help at all. But there's a lot of sediment, a lot of flooding comes in there. And we also know that there's evidence of some upwelling of the outer edge where it gets very deep. And it's postulated that might increase the plankton productivity as you go inshore, because you have nutrients in the upland. And that could be related to the pattern that we found in primates. There is a lot of interest in all those factors.
Yeah, thank you.
Well, I want to thank you guys for coming. Thank you, Dr. Dawn. Let's give her a hand.